Good morning, everyone. Thanks for coming. We have a very exciting session today on behalf of the Global Outreach Committee, challenges and status in development of gene therapies in low middle income countries. And we're honored to have our co-chair, Savita. She can...
Yeah, my name is Savita Rangarajan. I'm a consultant hematologist with an interest in hemophilia and hemophilia gene therapy.
And I am the co-chair, Richard Koya. I'm at Chicago. My interest is immunotherapy for cancer, basically T cells and stem cell engineering. Thank you. And our first speaker is Jennifer Adair. She is a principal investigator at Fred Hutchinson Cancer Research Center.
Thank you. Can everyone hear me okay? Awesome. Thank you for listening to me, whether that is in the room or online, or in the future virtually. I wanted to first just acknowledge that the land we're meeting on and the land that I get to practice my science on is the ancestral land of Indigenous peoples whose stewardship I greatly appreciate for giving me the opportunity to not just practice my passion, but to share it all with you. And I'm very excited to talk about access of gene and cell therapy in low and middle income countries, which really means access to patients everywhere. And I can thank Michael Luella, who's in the room with us and who you'll see me reference on several slides. Michael is an advocate for HIV cures that I know personally, and he's a great friend. He introduced me to this acronym REBSPA, which is the right of everyone to enjoy the benefits of scientific progress and its applications. This is actually the acronym that defines Article 15.1b of the International Covenant on Economic, Social, and Cultural Rights put forward by the United Nations. The United States of America signed this in 1977, but it's never been ratified. We are at a unique point in the gene and cell therapy field. We have taken off. You've seen it in this meeting. You heard it from our CEO yesterday. As of 2021, there are 536 different gene therapy developers. As of 2020, 373 registered gene therapy trials and $19.9 billion in financing for the sector. FDA Commissioner Dr. Scott Gottlieb predicted that by 2025, the US FDA will be approving double digits of gene therapy products per year. And it's no surprise that David mentioned at the George Stamm lecture yesterday that our society is the premier society because the United States has been responsible for almost 70% of the progress in this space. And while that's a fantastic achievement, lying on a lot of the laurels and the ideas of the people in this room, we're missing something really important from this pie chart, and that is gene and cell therapy in low and middle income countries. This is a World Bank's 2021 map of countries by gross national income, with low and lower middle income countries in purple and upper middle income countries in green. The red crosses represent countries where homegrown gene therapy is being researched and developed, and you could see that with the exception of India, we do not have a presence in any low and middle income countries. Over the course of this talk, I'm going to present to you two disease cases where this really matters. But first, I want to acknowledge Michael because I swiped this slide from his presentation on Monday. And if you get a chance to go back and watch the recording, it was in the session called Intersectionality of Society and Genetic Medicine. And it was entitled, Does Where You Live Matter? And I think this map kind of says it all. The two disease cases I'm going to make are probably not surprisingly for human immunodeficiency virus or HIV infection and sickle cell disease. There are tens of millions of people on the planet living with one of these two diseases, and the vast majority of them reside in sub-Saharan Africa. This is important for us in the gene and cell therapy space because a lot of the progress we've made has been in the development of HIV and sickle cell disease gene therapies. One of the first 10 gene therapies approved was Zynteglo. This is approved for the treatment of beta thalassemia, another red blood cell disorder, but it's applied in sickle cell disease as well. There were 7 clinical trials registered to treat sickle cell disease with some type of gene therapy between 2014 and 2021. And since 1994, 36 clinical trials using some kind of gene therapy to treat HIV infection have been registered. And if you just look around this meeting, there are 14 abstracts documenting some kind of gene therapy approach to treat HIV, and more than 20 actually documenting a gene therapy approach to treat sickle cell disease. But the number of gene and cell therapy trials for HIV or sickle cell disease that have taken place in Africa is 0. And I just want to let you sit with that for a second. I need to thank Dr. Julie McConney for giving me this slide idea. She's a collaborator of ours, and she also gave a talk in the intersectionality session on Monday entitled International Efforts to Expand Diversity of Genomic Data to Improve Health Outcomes. And I'm going to talk a little bit about the data that Julie referred to in that lecture because it is important for us to think about. The work she talked about was a culmination of more than 10 years of the Human Heredity and Health in Africa, or H3 Africa Consortium's efforts to put DNA sequencing of African genomes in the hands of Africans. There have been some really interesting findings that have come out of this work. For example, for decades it was thought that cystic fibrosis did not exist in Africa. It turns out that there is cystic fibrosis in Africa. The mutations are different than those that we've sequenced or documented in the United States or Europe. But the seminal work that came out of these studies is this paper, to me anyway, which published in Nature in October of 2020. In this study, they sequenced very deeply whole genomes of 426 individuals, including 50 ethno-linguistic groups across the African continent. They identified more than 3 million more mutations that were not described in any known human gene sequence to date, cementing once and for all that Africa is the most genetically diverse continent on our planet. They also identified 62 new chromosomal locations that are under strong selective pressure. Those were genes involved in viral immunity, DNA repair, and metabolism, which certainly also influence the outcome of gene therapy. I want you to look at this information not as a hindrance to getting gene therapy working. I want you to look at it as an advantage. A gene therapy for HIV or sickle cell disease that works in Africa will have the greatest transportability to anywhere else around the globe. And why is this important? Because we are not getting it right here domestically. This is a map showing you the prevalence of people living with HIV in our country, as shown by darker red. And the dots represent the places where homegrown gene therapies are being developed, and not all of them include gene therapies to treat HIV. So we ourselves are not getting our therapies where they need to go locally. Why is that? Because our current state of the art is complicated and it's expensive. Both HIV and sickle cell disease involve blood cells. In order for us to deliver a gene therapy successfully to blood cells, it has to go into the cellular DNA because those blood cells need to divide millions of times over the lifetime of the patient. And if we don't get it into the DNA, it will dilute out very quickly. This is typically done in International Standards Organization or ISO Class 5 clean rooms, which are incredibly intensive and expensive facilities to build and maintain. Now we have 10 approved gene therapies currently, and a large number of them, I think it's 7 now, are blood cell-based therapies. So what are those companies doing? Well, they're moving towards a centralized manufacturing model. So if you think about this, like most drugs you receive, there's a manufacturing facility somewhere that's receiving all the components used to assemble or formulate that drug, and then distributing it out to people wherever it needs to go. But in this context, the patient's cells are an important part of this process. So you have to collect the patient's cells locally and then ship them to that central manufacturing facility. And then once they've met release criteria, then you ship them back. Now, this might work in high-income countries. I don't necessarily think it's going to be sustainable, and I'll talk to you a little bit about why. Because the transport demand, the chain of custody, and stability burdens are not sustainable domestically if you look at the blood product market in our own country. And there are not blood product markets internationally, and so the thought that we're going to be able to ship cells from Africa to somewhere else to have this done and shipped back is just not realistic. In the next talk, you're going to hear my colleague, Dr. Sissy Kityo from the Joint Clinical Research Center in Uganda, talk about how this has not worked to bring other drugs to Africa in a reasonable time frame, or at all in the case of hydroxyurea to treat sickle cell disease. So we should not think that a more complicated gene and cell therapy strategy is going to work in this model, in this context. And it ignores the fundamental principle of why these therapies have so much promise. Because the patient is the therapy. The genetic modification is just the tool to execute it, but it's the patient's own cells that make this therapy work. And that means that we as a field and gene therapy as a treatment is uniquely positioned to bring access through local or place of care manufacturing. And most of the patients who have had access to some kind of gene therapy receive that through an investigator-initiated clinical trial at an academic institution or a research hospital. This fundamental idea of dealing with this inequity is the foundation of the Global Gene Therapy Initiative, also known as GGTI, which I had the pleasure of co-founding with Dr. Kitio, again, who you'll hear next, in October of 2020. The idea is the patient is central to accomplishing this treatment, and we need to think about bringing the therapy to the patient and not the other way around. We wanted to make sure that stakeholders across every sector, from basic research all the way through commercial development, were part of this conversation. And it all started when Dr. Kitio called me at 1 a.m. East Africa time, because she had just talked to another company about getting access to gene therapy who told her over the phone that it would take another 10 to 15 years before they could bring her a drug. And knowing all the information that I just shared with you in the beginning slides, she was very frustrated at the prospect of having to wait. She and I quickly aligned and identified a network of individuals, shown here as the founding members, who came together and said, yeah, we all have the same idea. None of us actually knows all the things it's going to take to get there, but we all want it to happen, and we're willing to work together to do that. Keeping patients at the center, we have an international community advisory board made up of patients and advocates. These include not just people who are living with HIV or people who are living with sickle cell disease, but people who are part of the Sickle Cell Network in Uganda or the African Academy of Sciences, people who understand the kinds of barriers that we'll face when we start to implement these strategies going forward, and it's the beginning of our community and outreach and education. We published our first working group report in 2021. We identified 4 pillars of activity that have to happen simultaneously if we are going to be successful in making this happen. We have to have community outreach and education early and often, or we won't get adoption when the time comes. There has to be clinical readiness to do an investigator-initiated clinical trial, and you need to build capacity because you have to have a pipeline of trained people that will continue that to have sustainability moving forward. You need economic stability, and that comes from technology development, which continues to make things simpler and cheaper as you go. And you need a regulation and policy infrastructure which supports public trust and ensures that you have a pathway to regulatory approval for commercial products. We have an ambitious goal to launch an investigator-initiated phase 1 clinical trial for gene therapy at 2 sites that have not done gene therapy before. One in India, the Christian Medical College in Vellore with Dr. Vikram Matthews, and the second at the Joint Clinical Research Center in Kampala with Dr. Sissy Kitio. The important thing about both of these institutions is that they are training institutions for other academic centers and research hospitals in the area, so the dissemination of information once it's developed will already be built into the system. We have an open source intellectual licensing strategy. This promotes global commercial development opportunities, and I'm grateful to Dr. John Tisdale of the National Heart, Lung, and Blood Institute, who has licensed his vector to treat sickle cell disease, and Dr. Boro Dropulich of Caring Cross, who holds the license for an anti-HIV duo CAR T-cell, which we will use in these initial clinical trials. And I want to take a minute to personally thank Boro, who is a founding member of the GGTI. Boro is someone who was in industry and became very frustrated with the lack of access and the lack of thought around how we could change paradigms to bring life-saving cures to patients. And so he left industry and formed Caring Cross, which is a 501c3 registered nonprofit. And it was critical for us at the time that GGTI founded because it gave us a home and a financial infrastructure with which to secure donations and actually execute on our initiative. So I'm just going to spend a few minutes walking through, very briefly, some of the activities in each of these 4 categories that GGTI is pursuing currently. In terms of community outreach and education, we have had a lot of activity, webinars, everything from a children's book to music videos by Moses Supercharger, an advocate living with HIV in Uganda, shown in the upper left, and the members of his Stigmaless band, shown in the upper center. We can't say what type of communication is going to speak to people, but local community members can. In terms of clinical readiness and implementation, we've been building the clinical infrastructure and capacity at both institutions. Vikram Matthew's group has done a tremendous job at CMC to launch their clinical trial. The picture in the bottom right shows the Prodigy device that they'll be using to manufacture CAR T cells there, and all of the paperwork for regulatory approval of the phase 1 trials submitted, and they expect to have it hopefully soon. On the upper right, you can see the apheresis and blood cell collection unit at the Joint Clinical Research Center in Uganda, which is gearing up for collection of cells from sickle cell patients. And on the bottom left is the hypothetical idea. It's an example. This is a portable clean room. So if we can't do things at higher than an ISO class 5 standard, we can create these portable ISO class 5 clean rooms or ISO class 7 with class 5 spaces where things can happen inside them in a portable fashion. And those can be moved from center to center so that we can greatly distribute the training and the access to these types of therapies. We're also training the first generation of African gene therapists. Dr. Lois Baiga is an employee of the JCRC who's spending a 2-year training fellowship in my lab in Seattle, and she is developing the process that we're going to use for the sickle cell disease treatment in blood stem cells. And this is done through a generous funding by the Bill and Melinda Gates Foundation as well as Caring Cross. And it's an important concept because our best chance for successful gene therapy to treat HRV or sickle cell disease in Africa is to have it made by Africans for Africans. And that sustainability is not just about creating new manufacturing processes. If you cannot diagnose and monitor the health of patients after you've given the gene therapy, you will not be able to understand whether or not it's working. I want to highlight 2 of our GGTI members and collaborators, Dr. Mohammed Draz and Dr. Umit Gherkin, both of whom are at Case Western Reserve University in Cleveland. Mohammed has built an assay on a chip that uses 30 microliters of blood to assay viral loads for HIV down to 100 copies per mil using a cell phone camera. This could put viral load measurements in the hands of the patients who own that information. Dr. Umit Gherkin has already made and commercialized the Gazelle device to measure hemoglobin and to define hemoglobin types using a paper electrophoresis cassette that can give you the result in less than 10 minutes for $5 a test. And there's a machine learning capacity built into this device that allows it to adapt to the hemoglobin variances in different populations. And we've already got this device placed at the Joint Clinical Research Center in Kampala. And none of this will move forward without the regulatory approval process. We're coordinating across all of the major regulatory agencies, the US FDA, the European Medicines Agency, the World Health Organization, and the new African Medicines Agency to ensure that we have a process that can not only ensure public safety, but can get us to commercial approval locally on the continent. I also want to highlight that the work of Dr. Kitio and the collaboration of the World Health Organization, in particular, Catherine Littler, we were able to get Uganda as a test case for implementation of regulatory infrastructure specific to gene and cell therapy. So with that, I just need to highlight all of the different people who are participating in this initiative alongside us. The representatives from each of these institutions volunteer their time. The only people paid in GGTI are the African scientists who are learning the training or are part of the development process. So this is an incredible initiative full of people who are incredibly passionate about making this move forward. With that, I'd like to thank you for listening.
We're going to take the questions at the end of the whole session. So moving next, we're happy to have Dr. Sisi Kitio. She is the Executive Director of the Joint Clinical Research Center in Uganda, and she's going to talk about our challenges of initiating gene therapy trials in low, middle-income countries. And she's participating in a virtual session.
Give a presentation on the challenges of initiating gene therapy trials in low and middle-income countries. From this slide, you can see that low and middle-income countries are generally excluded from gene therapy development. The distribution of the gene therapy clinical trials is mainly in high-income countries, and almost none in low and middle income countries. The current treatment for HIV is oral ART or combination ART. And we know that long-acting ART is already being scaled up in high-income countries. Also, broadly neutralizing antibodies as long-acting agents may be available in the next 1 to 3 years in these populations. Gene therapy will come in generations like we have seen in combination ART. And the timing of introduction of gene therapy cures will depend on how aggressively low and middle income countries pursue involvement in their development, including clinical trials, which, as we can see today, are currently not happening in these settings. Given that we need to pursue involvement aggressively, it is important to recognize that it took a whole 17 years for low and middle income countries to have access to combination ART from the time these drugs were developed. And this is shown very clearly on this timeline on the slide. However, we see that the timeline has shortened with regards to long-acting ART. It has taken about 5 years from the time long-acting ART was developed in resource-rich countries to these drugs being introduced in clinical research. Currently, a trial which started in 2021 is taking place in Uganda, Kenya, and South Africa. So the question we ask today is, how long will it take for a gene therapy to get to low and middle income settings? In order to accelerate this process, the Global Gene Therapy Initiative, also called the GGTI, was formed in 2020 to work towards enabling access and implementation of gene therapies as curative medicines in low and middle income countries, initially focusing on HIV and sickle cell disease. GGTI works by advocating for appropriate research, clinical development, capacity building, training, community adoption, regulatory pathway approvals, and also sustainability. So one of the main barriers to clinical trials in low and middle income countries is the high cost of gene therapies. This slide shows approved gene therapies in high income countries, their costs, and the numbers of patients who have accessed these therapies, even in high income countries, are still low. There's Integlo product, which could also be used for sickle cell disease, has a very high price tag. And this is beyond reach in low and middle income countries, as well as high income countries. So it does not make any sense to evaluate such products in clinical trials in populations where access will almost be impossible. To address this problem of cost, GGTI currently has 2 gene therapy products which we are implementing, and these will not be as costly as what I showed on the previous slide. One of these is a blood stem cell gene therapy product for the treatment of sickle cell disease, and this was made at NIH by Dr. John Tisdale. This is being licensed to Caring Cross, founded by Boro Kim and Rimas, and these are members of GGTI. There is also a T-cell gene therapy product, which was developed by Caring Cross, and this is a dual CAR T-cell for the treatment of HIV infection. We also have 2 members of GGTI, Dr. Els and Jane, who are currently developing technologies which could be first-generation delivery tools for in vivo blood cell gene therapies. Dr. Els has developed a viral vector which can deliver gene therapy to T cells in the blood. And this has since been licensed to Sana Biotechnology. Dr. Jane has developed a nanoparticle which can deliver CRISPR gene therapy into blood stem cells in the bone marrow. She has co-founded a new biotech company about 6 months ago to commercially develop this approach. So when you have the product, it has to be manufactured to provide investigational product for use in the clinical trials. And the Clinimax Prodigy by Miltenyi is currently the gold standard for gene therapy modification. This single device can automate all the processes and will not need a clean room. Also, we call this gene therapy in a box. However, if you look at this map, this is the distribution of Miltenyi offices in the world, and these are exclusively in high-income countries. And although there is a distributor in South Africa for the sub-Saharan region, the Prodigy is not supplied in sub-Saharan Africa. If it was ever supplied, several conditions imposed by Miltenyi on its sale and use create huge barriers for its use in this region and other low and middle income countries. So gene therapies are particularly hampered by manufacturing processes. And Professor Jane, who is a co-founder of GGTI, and Boro of Caring Cross, plus others, are working on simplified manufacturing technology using cheaper reagents and equipment, which can easily be placed at place of care in low and middle income countries. They're also working on mobile GMP facilities that can easily and quickly be deployed at clinics in low and middle income countries. Gene therapy trials require, like all trials, require coordination and communication within and across a multidisciplinary team. This slide has a list of the team that are required to conduct gene therapy trials in low and middle income countries. And it may not be completely exhaustive, but all this staff need to be trained and to also gain experience before they can get involved in gene therapy trials. We know that clinical teams with experience in trials are already available in low and middle income countries, but these need additional capacity building or reorientation to conduct gene therapy trials. And the training that is offered will depend on whether we have an ex vivo or an in vivo product for evaluation. So some of the areas that require training, which at this moment would have to take place in the US or other developed countries where gene therapy is already taking place, include establishing standard operating procedures for all study processes, leukapheresis to collect PBMCs, or hematopoietic stem cells for gene modification and cell mobilization, gene therapy product manufacturing and release testing, as I've already mentioned, screening of patients for the gene therapy clinical trials, conditioning of these participants, infusion of the product, and then clinical management of participants, identifying and managing toxicities, some of which are really unique to gene therapy or CAR T-cell in particular, and then management of these serious toxicities. Additionally, there is nursing care for study participants, We need to have QA/QC processes for everything that we do. And there are many new assays that are not currently available in low and middle income countries, but are required for these gene therapy trials. So research tests that are not done routinely need to be implemented, and also training done on those. There is a lot of community education, including counseling of the participants in gene therapy trials. And this is something that is completely new. In addition, there is need for long-term training in genomics, gene therapy, and basic science, so that basic science or development of gene therapy products can take place in low and middle income countries. And also reorienting biomedical engineers to support the new equipment that would have been introduced as part of these clinical trials. So one of our funding successes has led to a training partnership through Caring Cross, and this is funded by the Bill and Melinda Gates Foundation. The foundation has sponsored an employee of JCRC to train jointly in Jane's lab in Seattle and bring back her knowledge to train others at the Joint Clinical Research Center in Uganda. While in the United States, Lois, shown on this slide, is learning how to manufacture blood stem cells and T-cell gene therapy products for implementation of this process and training of others back home at site. We also have funding to allow one doctor and one nurse to travel to gene therapy clinical centers in the US to observe patients being treated with gene therapy. And this is just the start. We have, as I already showed, the clinical team that is established. All of them will need training in different areas. However, we also need to implement educational programs at our universities and medical schools if we want to continue to grow in this capacity. And we do not always have to take out our teams to the US, but we need to build that capacity in country, which can then train others. So some of the other gaps that hinder or prevent clinical trials from being implemented in low and middle income countries include apheresis. Apheresis is a starting point to obtain the cells that we need for gene therapy modification. Apheresis is not widely available, and it needs to be put in place, and also training has to happen. We have clinic space for other clinical trials, but we may need specialized space like clean rooms for infusion, and also access to advanced inpatient care to manage adverse events if this is required. We need to place several laboratory equipment that are not available and train on the new assays that are required for release testing of gene therapy products, screening and monitoring participants for safety and endpoints. Also need to be established for clinical trials. We need to have in place processes for cryopreservation, storage, shipping of some samples to specialty laboratories. And also, of course, it may seem obvious, but we need to have backup electricity to sustain and maintain the samples and cells that we are collecting. So for development to be truly successful, we must enable low and middle income countries to form their own intellectual property and new companies in the gene therapy space. Regulation and policy on biotechnology is missing in many countries. And working with WHO, local ministries of health and parliament. We need to support enactment of laws to support gene therapy research, product development, and also its approval for the different countries. And then most importantly, we cannot develop gene therapy without the patients who need it. So community outreach and education is central to informing and engaging the public and addressing myths about gene therapy, while at the same time educating them about the benefits. We already have existing advocacy groups who were very active in accessing HIV treatment and can still be utilized and can be allies in development of gene therapy for the cure of HIV. Understanding how to communicate and what patients need are critical to the success of our programs. So those are some of the main challenges, but as we work around them and solve them to enable gene therapy access in low and middle income countries, we will at the same time be advancing gene therapy for everyone, everywhere. So for us to do this, we need partnerships. We cannot do it alone. The expertise that is available right now is in developed countries. We need funding to be able to do this. And partners that we are working with include the NIH, who is working closely with Bill and Melinda Gates Foundation. And as you can see the roadmap, this is to have these kind of cures available in low and middle income countries. The other partners that we are working with include HIV Cure Africa Acceleration Partnership, which is called HCAP. And these have been working in different capacities, even to determine what do we need as a gene therapy, what are the qualities, or what should a gene therapy look like if it is going to be globally accessible. So we thank all the partners. Many are not mentioned here, but these kind of partnerships are the ones that are going to propel us forward and be able to both conduct gene therapy clinical trials in developing countries, but also increase access in these populations. So I would like to thank everybody for your attention. Thank you.
Thank you, Richard. We've heard in the previous two talks about the desperate need for developing gene and cell therapies in LMICs. India is an LMIC, but it's also been nicknamed as the pharmacy of the world. It is my pleasure to introduce Mr. K.V. Subramanyam, who is the president of Reliance Life Sciences, which is a very large life sciences company based in Mumbai. It is foraying into gene and cell therapy for the very reason to make it cost-effective for LMICs. KV.
Thank you, Savita. Thank you, Richard. Good morning, ladies and gentlemen. It's a pleasure to be here and to talk about manufacturing barriers and solutions in the context of gene therapy in low and middle income countries. Before I do that, I would like to spend a short while on Reliance Life Sciences and our cell and gene therapy initiative. So Reliance Life Sciences is part of the Reliance group of companies, which is the largest private sector enterprise in India. Our business is essentially in medical biotechnology-based products and services, and used in critical and intensive care. So you will not find products in the chemist shops. We are a research-driven company. Almost all the products that we have in the market have been developed in-house. We are fully integrated in the value chain from research, animal studies, manufacturing to marketing. I have a subsequent slide which talks about the various initiatives, but we were the first to have the largest integrated plasma proteins facility in South Asia, in India. We have the largest portfolio of biosimilar products in development. And the reason I brought in biosimilars is there are parallels between development of biosimilars and cell and gene therapy, which I'll talk about. And we maintain very high quality standards. Several of our facilities have been approved by US FDA, MHRA, EMA, and so on. The main campus is near Mumbai. It's in Navi Mumbai, a 20-acre campus. We are currently building a much larger campus, 8 times the size of our current campus. And we have about 1,200 employees. So this is a snapshot of all the initiatives that we have. So we have 3 main businesses in plasma proteins, biosimilars, and later version generic oncology pharmaceuticals. We have an internal clinical research organization. We also have a small initiative in regenerative medicine. But at this time, we are focusing on 3 areas. One in building our next generation diagnostics business in India. We're also building a vaccines initiative, both human and animal vaccines. And as far as innovative technologies are concerned, we are at this time building a cell and gene therapy facility apart from early stage work in mRNA and oligonucleotides. And as I mentioned, our whole theme is to do things in a very integrated manner, right from research, preclinical development, clinical development, quality management to manufacturing and marketing. So as far as gene therapy programs are concerned, the same integrated approach applies here as well. We are currently building a pilot scale manufacturing facility in our existing campus, encompassing plasmids, AAV vectors, lentivirus, and CAR T cells. And we are contemplating having much larger commercial scale facilities in the new campus, which will be ready in about 2 years' time. By that time, hopefully, we'll have at least a couple of gene therapy products developed and hopefully approved. So in terms of our programs, we have 9 gene therapy products that are currently under development, and we have 7 CAR T cell therapies under development. I will not go into the details of each of these cell and gene therapy programs that we have taken up. So it's a fairly broad-based development that we have taken up. So much about Reliance and the gene therapy programs. Now I'll turn my attention to the challenges for low and middle income countries. Today, if you look at the current paradigm of gene and cell therapies, it's a high price point to patients and very low accessibility. Now, one has to change that paradigm to having prices very low or competitive and much greater access to patients. And today, if you look at the world, in the low and middle income countries, there are only a few countries that are working on gene therapies, India in particular. But the other countries are Brazil, China, Russia, South Africa, Thailand, and Turkey. So the development objective for us, as far as gene and cell therapy is concerned, and for that matter, other biopharmaceutical products as well, is to have an expansive range of products and get that to most people who have issues about affordability. So it has to have a very high social impact. So the development paradigm, therefore, boils down to saying that we need to have high quality and safety at very affordable costs. I just put in a number of $25,000. And if a gene therapy product is targeted at less than that price, I think it'll have much wider applications. We should also, while talking about LMICs, not forget, apart from the socioeconomic profile, that the insurance penetration is very low. For example, in India, about 95% of the patients typically spend from their own pockets. So we don't have a very evolved insurance system. So that's all the more reason that we need to have high quality products with good safety being delivered at very competitive costs. So that becomes the development paradigm. So how can manufacturing contribute to that development paradigm? So if you stack up the costs involved in manufacturing, one element is the innovation cost that is amortized. You have all the manufacturing expenses, consumables, utilities, and so on. Capital charges become a big component of the cost because these are capital-intensive investments that you make across the value chain in cell and gene therapy. And then, of course, the people costs and overheads. But within that, the manufacturing expenses and capital charge is where we can make significant contribution if you get your manufacturing right. Manufacturing focus also enables quality and consistency and also bring about larger scale where you can essentially take care of more patients as far as the market is concerned. So in essence, manufacturing plays a very vital role in enabling competitive cost, quality, and scale. But then there are several barriers as far as manufacturing is concerned. So first is technology, both in terms of access and absorption. So even if technology was available and you're able to access technology, you also need to have reasonably trained people on the other side of the equation to be able to absorb that technology and deploy them productively. The other barrier is in project management capabilities as well as in operational capabilities. It's a question of competency. And then you have the big issue about talent, not only availability, but also inclination. And the reason I put the word inclination here is that today we are in a very strange situation in India because a lot of youngsters, for them, manufacturing is a less fashionable place to be in. They're more attracted by the technology companies, and we have a large number of startups that are happening in India, so it's becoming that much more difficult to attract a lot of young talent into manufacturing. And here again, the aspect of competency development comes into play. Capital is another barrier because these are very capital-intensive investments that you have to make. So there is also a question of availability of capital as well as high cost of capital. Typically, interest rates are much higher in our part of the world. And then, of course, a lot of methods development that needs to happen along with the development of cell and gene therapies. And then we have the regulatory environment. We need to have regulators who are far more educated on the intricacies of cell and gene therapy and be able to support its development. So these are the main barriers. So then one would naturally ask the question as to what are the solutions to address these barriers. So now I've listed some of the solutions that one could think of, and this is something that we have been working in Reliance. One is, of course, on the technology side, partnership with academics as well as NGOs. And we can also access or develop technologies in areas where there is freedom to operate from an intellectual property point of view, and then also take recourse to support of gene therapy societies like ASGCT. From a project management perspective, and this is coming out of our experience, we can use EPC companies who have experience in the Western world in developing facilities and also take recourse to expats, which is what we have done in a few cases in Reliance. And when we started Reliance, we recognized that talent was our biggest challenge, so we instituted our own small institute to develop in-house talent and tied up with universities for our employees to pursue master's and PhD programs on problems that face us. As far as capital is concerned, there needs to be mission-driven funding, and at least for us in the Reliance system, we've had promoters who were very patient with us in terms of the long development and gestation period that it takes, and they did recognize that this is a mission that they need to fund, and so where you have mission-driven funding is something that can address the capital barrier. And on quality management, also the question of partnerships with academics is one approach that you could adopt. And as far as regulatory aspects are concerned, there are quite a few interdevelopmental FDA training programs which are also happening in the existing areas of both pharmaceuticals and biopharmaceuticals. So that is one way you can address that barrier, and also interaction with a lot of international agencies. Just put PICS here as an example. So there are several solutions that one could take recourse to address the various barriers that come up as far as manufacturing is concerned. So I thought I'll also portray the biosimilars parallel. If you look at what we have done in biosimilars, we currently have 21 products in the market, which is the largest for any single company in the world. And we export this to India and rest of the world countries. We are also now working on a few products that can go into US and Europe through a clinical development program. We have another 15 products under development in different stages. We have built manufacturing facilities. We have 2 facilities for microbial with microbial fermenters, 2 facilities on mammalian cell culture, both stainless steel and single-use bioreactors. We have 2 facilities for fill finish, covering all the way from pre-filled syringes, vials, lyophilized vials, and cartridges. So the development paradigm that we adopted here, which we think is equally applicable to developing cell and gene therapies, essentially focus on high quality at competitive costs. And so the strategy that we adopted was to develop virtually all products in-house. We have put together a very competent team as far as cell and gene therapy is concerned. We have the capability to do clinical development in-house because we have our own clinical research organization. We've also built capabilities to develop bioassays. So the important aspect here is that once you have your own manufacturing and once you have your own clinical development and bioassays development, then the costs are that much lower. The time to market is also much faster. You know, it's very difficult to imagine if you didn't have this, you'll have to be queuing up with a contract development and manufacturing organization or an external clinical research organization. That would take much longer time, and it would drive up the cost significantly for us. So we think that the gene and cell therapy programs of Reliance Life Sciences can take lessons from the biosimilars, which has been pretty successful in the Reliance scheme of things. And that's how we are planning to build this out, within which manufacturing is a very significant component. So that's basically what I wanted to share. I thought I'll quickly sum up the take-home points. The first is to say that the development paradigm must be looking at an expansive nature of products and services, and which are very impactful. And you can do that if you have high quality and safety at very affordable costs. Manufacturing plays a very critical role in bringing down costs, quality, and scale as long as you get it right. And then there are several barriers that not only in India but in other low and middle income countries face in the context of manufacturing. And there are solutions, and a big part of that solution is one is partnership and the other is competency development. And then we think that the biosimilars play that we have is a good learning as far as developing a broad range of cell and gene therapy products and services are concerned. So in the final analysis, what I think will drive home these initiatives will be a number of partnerships. Fortunately, in Reliance, we've had a tradition of a number of partnerships, not only with other Indian pharma companies, but also with countries outside the country in terms of out-licensing some of our products. We have co-development partnerships with companies in Europe and US. So I think partnerships is the way to go in expanding footprint of cell and gene therapy products and services. So thank you very much for your attention.
Thank you. And then to our final talk before the session for questions, I would like to introduce Dr. Mike McCune, who is actually going to be virtual as well and is at the Gates Foundation. And he's going to talk about bringing safe, effective, and accessible gene therapies for HIV and sickle cell disease to low and middle income countries. Thank you.
Hi, it's a pleasure to have the opportunity to talk in this session. I'd like to thank the organizers for the invitation. In the next 20 minutes or so, I'd like to give you an overview of a program that has been set up at the Bill and Melinda Gates Foundation called the HIV Frontiers program, which is designed to bring safe, effective, and accessible gene therapies for HIV and also sickle cell disease to resource-limited parts of the world. By way of some background, for those of you who are not steeped in the current situation of the AIDS epidemic, there are now some 38 million people living with HIV worldwide. And even though we've had antiretroviral therapy for the past 30 years or more, almost half of those are living without able to gain access to or to maintain themselves on antiretroviral therapy. And many of those live in sub-Saharan Africa. And the goal of the program that has been set up is to find a way to bring an effective, durable, safe, accessible, affordable, curative intervention for HIV to all of those people and to everybody else if we can pull it off there. And looking at the dimensions of the epidemic and the pandemic in sub-Saharan Africa and other low and middle income countries, where there's a very high incidence of disease and lots of transmission, poor infrastructure, and also the usual stigma about going to take advantage of that infrastructure, which is available. What we imagined from the get-go was that any curative intervention had to be very simple. A single shot, euphemistically called, administered point of contact, percutaneously, simply without much in the way of intervention. And boiling that down to a construct that might work, we're thinking about in vivo delivery of a genetic manipulation that might lead to antiretroviral-free viral suppression. Other parts of the target product profile for such a cure, of course, include the fact that it has to lower the viral load to a point where the individual who's infected with HIV doesn't get sick and also is not likely to transmit the virus to another individual. We want the duration of the remission to last at least for 3 years, which is longer than any long-acting antiretroviral that is available today. Modeling it out, it's quite clear that unless the intervention prevents or controls reinfection, that all the gains that are lost with respect to reduction in incidence will be all the gains that are made will be lost. In the pessimistic scenario, you can read through this paper here, which is the likely scenario, will be back to baseline without that kind of prevention of reinfection. We need to have a test kit, by and large, which can be used to understand whether the viral reservoir has been reduced. We also need that as a home kit to understand whether it's come back. And that's something that needs to be in place before we bring it into these high incidence areas. And not least, it's got to be safe and affordable. And we're pegging this out probably to be cost effective with HIV antiretroviral therapy on the order of $1,000 to $2,000 in sub-Saharan Africa. And again, what this is a sustained art-free suppression, a functional cure of HIV, not eradication. It has to have an immunologic effect that will keep the virus at bay over time. It's a tall order. How do we get there? We set out the work from the beginning to be divided into a number of bins. One was we needed to understand what the design of this intervention might be for HIV. We still do not know that. There's some ideas, but there's a lot of work ongoing now, and we hopefully will know it soon. But in the meantime, in order to make it durable as an in vivo modification, we've decided to focus on sickle cell disease and modification of hematopoietic stem cells for treatment of it, imagining that if we could do that, that later we might introduce interventions for HIV also through the stem cell or other long-lived cells in the hematopoietic system for suppression of viremia. With respect to detection, we know that we need to detect circulating biomarkers that are hopefully non-viral of the rebound competent reservoir. And to do this, we've set up, and I'll describe very briefly, a 5-team, 40-lab, 4-continent reservoir consortium to dive deeply into the biology of that reservoir. We know that we have to deliver this into parts of the world that have never seen gene therapy before, and we need to know how to do that, and we need to start that sooner than later. So we've already set up a public-private partnership called the HIV Cure African Acceleration Partnership to engage key stakeholders now so that we design cures that can work and that will be accepted later. And finally, we knew from the standpoint of the foundation that we couldn't do this alone and that we needed to engage partners along the way that could bring in orthogonal competencies, resources, and by and large, just share the risk. From the beginning of the program, which was launched officially in early 2019 with resources from the foundation, we began to reach out to the NIH, in particular NHLBI and NIAID, but also the Office of the Director, to form a collaboration with them that was meant to be one-to-one in the beginning, but which has been far exceeded by the NIH now, focused on in vivo gene therapies for HIV and sickle cell disease, not including all of the other work done by the NIH and by the foundation on sickle and HIV, but focused exclusively on that. These funds are going out by and large in the way of grants to academic and non-academic institutions, but we've also taken advantage of the resources of the Foundation for Equity Investments and made a number of those into companies that are working now on cure interventions, including Immunocore, BioNTech, and Vir. Another model where companies are putting skin in the game is one that we've set up with Novartis at the Institutes for Biomedical Research, in which they put in money and we put in money to stand up a research team within the hallways of NIBR to work on in vivo gene therapies for sickle and HIV. And finally, we are looking for, once we have biomarkers available, a multinational company that can help us to put that into a form that can be taken onto the field. Where are we today? We've identified a number of different candidate vectors that can target hematopoietic stem cells in vivo. I'll show you very briefly some data related to that. We've made, I think, substantial progress in understanding the biology of the rebound competent reservoir. We've engaged key stakeholders in sub-Saharan Africa, and we've formed these multiple partnerships that I just showed you. And actually, after 3 years of work, we've narrowed it down not to one, but to 2 ways in which a single shot intervention might lead to sustained antiviral suppression. One of those is targeting and editing of long-lived cells in vivo. This, as I mentioned, would be applicable to sickle cell disease. Another that I'll talk about briefly relates to therapeutic vaccination, not applicable to sickle, but something that could be given to individuals who are on antiretroviral therapy and lead to an immunologic response that would suppress their viral load. With respect to the first platform, very briefly, we want to move from ex vivo therapy to in vivo therapies shot across the arm. We want to hit for sickle and HIV self-renewing hematopoietic stem cells that are multi-lineage of potential. For HIV, we are also funding research to target T cells that are long-lived, like T stem central memory cells and B cells in vivo. To do this, we've had to confront a number of obstacles that relate to in vivo therapies. One, we think probably the most important one right now is just targeting them in vivo selectively without a lot of off-target hits. Once we can do that, we need to bring in editors or things that can insert new genes into those cells in such a way as to create cures for HIV and sickle. For the stem cell, it's likely that we'll have to work through various maneuvers for conditioning and mobilization. And at the end of the day, we'll also have to figure out how to manufacture all this and bring it safely onto the field. For now, we've been focusing on these vectors, viral and non-viral. We've made a large number of grants. I show some of them here over the past 3 years to a number of academic as well as corporate partners, including the multinational company Novartis. And in blue, I show ones actually that are beginning to show us promise as ones that could work in vivo. One, for instance, is a helper-dependent adenoviral vector that's been championed by Andre Lieber and his colleagues at the University of Washington. It's a vector that's been used around the world for many years. It's easy to produce. It has a large cargo size, and it can be modified to have fibers that bind selectively to hematopoietic stem cells, which is what he's done. And if those cells are then mobilized in the peripheral blood, they can be hit and modified. The cargoes that he's used to date include the Sleeping Beauty transposase and the human gamma globin gene to transduce hematopoietic stem cells and ultimately red cells in non-human primates. And on the bottom left, you'll see if 20% correction is thought to be suitable now for clinical benefit in the context of sickle, animals now are hitting up to 80% of correction, well above that needed for long periods of time. Andre's also worked with David Liu recently to use prime editors in the context of sickle cell disease and has shown at least already in the sickle Towns mouse, that prime editors are capable of correcting the sickle beta globin gene to one that's wild type. And this is something that's now moved into non-human primates. In addition to this, for HIV, Andre has shown that you can introduce an OND antagonist called ECD4, which basically inhibits every HIV and SIV isolate known at 1 microgram per mil. And on the bottom left, you can see there's high level expression well above that 1 microgram per mil for months on end after transduction of hematopoietic stem cells with this vector. And on the bottom right, this is a challenge in which increasing doses of Shiv88 are given to that animal to show that there's only breakthrough infection out to 890 nanograms, which is log orders higher than what has been shown in previous experiments using ECD4. Another set of very clever experiments have been carried out by the team at Intellia. These were reported last year at Keystone. They've moved much further since. They've modified lipid nanoparticles basically in such a manner that they're tropic for hematopoietic stem cells in bone marrow. And once introduced in vivo into wild type mice in the bottom left, can edit hematopoietic stem cells in the light blue well above the 20% threshold for clinical benefit. And on the bottom right, using humanized mice, hematopoietic stem cells that are human can also be modified by this vector selectively above that 20% threshold. At this point in time, we're moving as many of the vectors as we can head to head through a humanized mouse core that's been established by Stefan Radke at the Hutch, so that all can be compared with data sets that are derived basically from the same animal model, hoping to down select this year a small number from the 40, probably no more than 4, that can be moved into a non-human primate model that's been set up by Hans-Peter Kiem at the University of Washington Non-Human Primate Center, meant to be GLP-like IND enabling. And these experiments will help us to move forward with other metrics in mind. One is not only to have vectors that have favorable on-target effects, but also ones that are safe, that can be paired best with the best editors that have the right cargo size, for example. We need to understand which ones are most likely to be developable and brought into LMICs and other resource-limited parts of the world. And importantly, as I'll stress again towards the end, we have to have rights for global access so that these products can be brought to people everywhere. The second platform I'll talk about very quickly relates to therapeutic vaccination. In one arm of the work that we're carrying out, we're looking at highly networked epitopes that are thought to be recognized by individuals who are able to control HIV on their own, and to put that into a polyepitopic mRNA vaccine that could be delivered to individuals that are on ART. Another approach that we're following is to look at the ability of antigen antibody complexes to be taken up by Fc receptors on antigen presenting cells and to secondarily induce an immune response called a vaccinal effect. And we've just made a large investment into Vir that I mentioned earlier, the company Vir, so that the mutation GALI-E can be introduced into such antibodies to enhance their potency. The next steps as we see them going forward are to take all the information that we have to date, that we've gained, that the NIH has gained, that others have gained, and to down select, at least from our purposes, 6 programs moving forward in the next 4 years, 4 of which will be focused on at targeting and editing hematopoietic stem cells, and 2 on therapeutic vaccination. We imagine that some of these will go forward and others not. And at the end of that period of time, we will down select another 2 to 3 of them to move forward to the clinic. And thereafter, we will hope to see in clinical trials the extent to which these vectors are efficacious and safe. I'll briefly say that we have set up this effort to understand more about the biology of the rebound competent reservoir. The reservoirs consortium is working now. It's about 2 or 3 years in. A lot of data have been gathered, and we hope that these efforts will lead to a kit at the end of the day that can be used to detect the rebound competent reservoir. Capacity building is very important. You've heard about that from the previous speakers, and we are firmly behind all the efforts that are being carried out by the Global Gene Therapy Initiative and Caring Cross and others with respect to ex vivo gene therapy in sub-Saharan Africa. But there's a lot of work to do. And that's what we're hoping to work with them and others on in the near future. In summary, we have identified these candidate single shot interventions that we are working on to move forward for proof of concept in non-human primates and then in humans. We do think that we can use these to edit hematopoietic stem cells and other long-lived cells in vivo. There is an effort ongoing now to identify circulating non-viral biomarkers, and these partnerships are growing and moving in a way that is cohesive and I think likely to prepare the field for these interventions should they come to bear. There are obstacles to address, and I just want to highlight these for you before I end. One is that there's a lot of effort now to do gene therapy, carry out gene therapy, but most of those efforts are aimed at diseases other than HIV and sickle cell disease. And we're hoping to use the resources of the foundation and the NIH to move that needle so that more and more work is done on those diseases that are so prevalent outside of high-income countries. In so doing, we need to gather global access rights for the use of the funded developments that are made. We need to have a better definition of the design of a single shot cure for HIV and understand whether those for HIV and sickle are safe. We have to also bring down the cost of goods. I show this diagram to show you how, at least for monoclonal antibodies, that cost has been brought down some 50-fold over the past several decades, primarily because there was a need to do it. And I think it's the same thing is going to happen for in vivo gene therapy. If there is a way to do it, we have to figure out a way to do it affordably. And not least, we have to understand biomarkers and engage stakeholders in LMICs so that these therapies can be accepted when they're brought to bear. We think now is the right time to push forward, and I'll end with just some thoughts to say that we have right now the advances in gene therapy, which enable us to do this. And so now is the time to move. We are understanding more about HIV and its biology, something that we've known for a long time with sickle. We're catching up with HIV. And so now's the time we can actually think about discovery of biomarkers. We've learned over the past few years with COVID that transformative technologies like mRNA vaccines can really make an impact. And people in multiple societies understand that now. But I think it's also been clear over the past few years that not everybody is able to take advantage of those technologies. And that's where we have to move. And that's where at the foundation and elsewhere, we're trying to make sure that funded developments are accessible to everybody, everywhere. So I'll stop there and look forward to any questions that you might have. Thank you very much for listening. Bye-bye.
Thank you. Any questions from the audience over here? I would like to say that Jen Adair will be there in about 2 minutes. And Mr. K. And Mike is there virtually as well.
Good morning. Thank you for this great set of presentations. Cell and gene therapies, this is why we're here. These are all very, very exciting, but they occur in the background of a larger medical question, such as viral suppression for HIV, and something as simple as hydroxyurea for sickle cell disease. So I wonder if my colleague, who's one of the largest manufacturers of biosimilars in the world and small molecules, how do you see marketing of hydroxyurea and assessment of sickle cell disease status going hand in hand with cell and gene therapy for sickle cell disease?
Mike, can you take the question?
That was actually for Dr. Subramanyam, but it's okay.
Oh, we don't make it.
Yeah.
So if I understand your question correctly, talking about sickle cell anemia and hydroxyurea, that's an area where we are not participating. So I think Mike or somebody else can take that.
What could I do to convince you to make hydroxyurea in and for Africa?
I beg your pardon?
What could I do to convince you to make hydroxyurea in and for Africa?
I didn't.
What can
convince?
How can you be convinced to make hydroxyurea?
Okay. So we are very focused on biological products and services. So it has to be within our product portfolio strategy. So that's, if it is fitting with that, then that's something that can convince us, and the market opportunity has to be significant. We are very clear that we are not looking at big profitability as far as these areas are concerned, particularly in gene and cell therapy. So
Okay, so your answer was marketing availability.
yeah.
Dr. McCune, you had an answer as well?
My answer is to agree with you that we need to figure out a way to do it. But do I have a pathway by which that can occur? Well, there have to be incentives. And the incentive structure right now for bringing medicines, even in the United States, as Jen pointed out, right, is mismatched with the need. So your question has to be answered, and it has to be answered in a way that works. And it has to also be brought to bear that the progression of events that you've heard in this session will occur over time sequentially, right? So with the NIH, for instance, we have a collaboration to look at in vivo gene therapy, imagining that that's a ways off, obviously, but we don't want to stop the work to bring drugs like hydroxyurea, or for that matter, good antiretroviral therapy to all parts of the world. So I think it's just something that we have to push on.
Great.
Yes, thank you very much to the organizers for putting this together. It's something we do need to be talking about more. So my question, I don't know, is this mic on? Yeah. Okay. My question is a little more detailed, because we've actually gone into India and we did a clinical trial, one of the first clinical trials we've done with CMC Valor for hemophilia, and we've actually brought in an AAV and lenti-based gene therapies. And some of the major hurdles that we had to overcome, one was actually IP, getting intellectual property rights into India for first-in-people trials. So doing this is a problem, just getting IP. Secondary is the regulatory aspects within each country. So having regulatory hurdles within India was a huge problem, and it took us 7 years to actually work through the regulatory aspects to get our gene therapy approach approved. And we're now, in fact, today we're treating probably the first patient. We've mobilized cells and transducing stem cells at CMC Valor. So between IP, between regulatory aspects, and the cost of goods, which getting down to $1,000 for a gene therapy is a Herculean goal compared to what it's costing today. Not that it can't be done, but these are formidable hurdles, and I'm wondering if you're thinking about some of those aspects of IP is going to be a problem. So what do you think of IP? I know that Reliance, you mentioned knockoff drugs, but these aren't knockoff drugs. There's still a lot of years on some of these patents. So how are you thinking about that?
Yeah, I'm sorry I can't see who's speaking, but I applaud you on your efforts, and congratulations on getting into the study in India. Sure, I mean, IP is incredibly important, as we've seen in the COVID-2 vaccine and the rollout there, shambolic as it has been. We at the foundation require that anything that we do in the beginning is done with a partner that provides non-exclusive rights for global access. And if they don't do that, then we don't fund. If they do that, then we have to provide incentives for that corporate partner, if you will, to move forward in the way of downstream implementation. And that's something that the foundation has been doing for a number of years now. The platforms that we're talking about are applicable not simply to HIV and to sickle in the case of gene editing, for instance, of hematopoietic stem cells, but to multiple other diseases, indications in multiple other countries. And we ask for those non-exclusive rights for those two indications in LMICs, leaving a corporate partner the ability to use the same platform for other indications, or even the same indications in high-income countries. So that's number one. Number two, the cost of goods, is likely to be, as you say, a Herculean effort to bring it down to $1,000. One way to do it is to have tiered pricing, where a corporate partner may charge one price in a high-income country and a lower price in another. That's done actually routinely. The other, from the power of science, I mean, you've seen over the years now, the cost of sequencing has gone down. I mentioned in my talk the cost of making monoclonal antibodies has gone down. And we may not reach 1,000, but I'm pretty sure we'll be able to bring the cost down well below the $2 million price tag for now for ex vivo gene therapy. So, regulatory considerations also, of course, have to be considered. And we are thinking about this now. We're convening, excuse the noise in my background, people together from sub-Saharan Africa today to talk about regulation of in vivo products or ex vivo products tomorrow.
Okay. Oh, good to know.
We can, I think, you can talk about the regulatory problems in India.
I can add to that. We talked about three things. One is IP, second is cost of goods, and the third is regulatory. From our point of view, we are very clear that we will operate only in those areas where we either have freedom to operate or we have in-licensed. For example, in the biosimilars program, we have several, we have high expression cell lines which we have licensed in. As far as cost of goods is concerned, okay, initially the cost will tend to be high, but from our experience, as you do more work and you gain experience, you produce repeated batches, you can get the cost down. With scale, your cost can go down. And also, when you use much better expression system, which can give you high yields, that's the other way in which you can get costs going down. And one of the other important aspects is if you get your capital costs right, you know, from day one, you have a competitive advantage as far as costs are concerned. So that's as for the cost of goods are concerned. As far as regulatory aspects are concerned, I think now there is greater awareness among regulators. They're much better educated as far as cell and gene therapies are concerned. And I think it should take us much lesser time as it used to be compared to what it used to be before in terms of getting these products approved.
Hi, and thank you everybody for some great talks. You know, I guess one of the questions I had was the role that we can play here in the audience of trying to advocate for things. And, you know, we saw some slides how the NIH has contributed to some research here. But, you know, manufacturing costs are something which, again, encouraging the NIH to support decreasing research around manufacturing and bringing some of those costs down. But also, you know, efforts, for example, in HIV, from a clinical perspective, have relied on USAID and PEPFAR to do this, and those fundings have been decreasing in recent years. Do you have any advice maybe for folks here in the audience of how they may advocate for continued support for development here beyond what the efforts we heard today?
Use your voice, vote. I mean, there's organizations such as this have individual as well as collective strengths, and we can't be silent. I think a good example, I mean, for those of us who grew up in the early days of the AIDS epidemic, we were pushed by the community, and the research community actually was taught by the community of patients and their care providers and their families, and that really made an impact in the United States. Sissy can talk about the advocacy in Africa that brought eventually, but you said 17 years, Sissy, right? But eventually, with your voice, there is more antiretroviral therapy in Africa than ever before. There still needs to be more. The sickle community has had less cohesiveness and advocacy, and I think, you know, this would be, again, something that needs to be engaged. And the voices in the room are both ones to start a discussion and also to listen. And I think this kind of exchange has to happen on a broader scale, and it needs eventually, as it was in the AIDS epidemic, politicized and brought forward in a way that is noticed and that is embraced.
Yeah, maybe I can add, I think we have to also learn from the history of where we have come from and how we got antiretroviral therapy in developing countries. It was partly due to advocacy, both in the US, very active advocacy groups in sub-Saharan Africa. There is a special group in South Africa. So, but it was continuous. It was continuous advocacy and talking about the issues, and also talking to very high levels of even in the US administration. So I think we are riding on that, and the advocacy groups for especially HIV, and I think, as Mike said, for sickle cell disease, maybe we need to actually combine efforts and work together. Otherwise, these same groups that advocated for treatment to even get to low and middle income countries should still be mobilized. And I think some of them are working with us to see how we can get HIV cure to these patients. In particular, I think we should also focus programs like PEPFAR that are providing treatment for HIV, because if we had, looking at even cost effectiveness, if we had a cure today, then we would need to spend eventually less money within the PEPFAR program to provide antiretroviral drugs for everybody for a very long time. And I think, so this, we should also be appealing to these funders in terms of this kind of data on cost effectiveness and what we prevent, because ART for now is given forever. But I think we need to use synergies between sickle cell and HIV, and we advocate as a group. You did talk about regulatory, and I think if it took that long in India, maybe there are also things to learn from that. But right now, the WHO has been working on documents which are out now, guidelines on human gene editing. And these are especially important for low and middle income countries where there is no regulatory today. There was no need for the regulatory to be in place, but given what is happening internationally, the regulation has to come, and now we can be guided by WHO. So there is this provision, and WHO has already agreed to use Uganda as a test case, at least for sub-Saharan Africa. So I think also part of what we need to do is to educate our regulatory bodies, because gene therapy is new to many of us, and we need to educate people so that they understand what we are talking about. And so that is part of the community education, which we are also engaging with, which community includes also regulatory bodies as a stakeholder, because they have to understand before they can even apply any policies that we build. And also the other stakeholders, of course, are politicians. They're the ones that really pass our policies in the country. But at least we have a good safety net now with WHO that has guidelines that we can follow or we can use.
So, is this on? Oh, perfect. So in terms of fundraising, I want to highlight how much we can learn from the HIV space. PEPFAR, USAID, those were a culmination of strong advocacy efforts by the patients themselves. And I think it's poignant to note, a lot of us think about when advocates stormed the NIH, right? But there was another very important demonstration that took place here in Washington, D.C. If you are interested in learning more about it, I highly recommend the podcast Radio Lab, and the specific episode is called Ashes on the Lawn. This was a demonstration where the loved ones, people living with HIV and their partners, came and threw the ashes of those who had died, whom they loved, on the White House lawn. Because that is what it meant to them for our government to be ignoring the fact that there was not a cure. Now, this is not to say us sitting in this room who may or may not be patients with these diseases, don't have a place in this. Our job is to include patients in the conversation to make sure that we're developing the correct therapies. That will inspire them to say, why aren't we getting these? And the public demand is what will create the funding. So if you are looking for something to do right now, practice communicating and reaching out and start including patients and patient advocacy groups in the work that you're developing.
Thank you. That's a very good point. Can I have the next question?
Yeah, hi. My name is Chris. I work for a pharma company, and I know pharma companies have probably the reputation of more getting in your way than being helpful. But I'm an engineer, so I work in a lab or in a manufacturing setting, and me and my peers, we'd love to help you guys out. And so it's not the technicians and the engineers and the scientists, I think, who are getting in your way. But my question is around the getting the cost of an in vivo vector down to $1,000 to $2,000 per dose, which I think is something that is possible. But a big lever to enable that is volume, and like how many doses over a fixed period of time you need. And the more that we would have to make, the more likely we would be able to get down to that $1,000 to $2,000 per dose. So I'm curious from your assessment of like the patient population and the ability to distribute these drugs, what would be the volume that you would be looking at, kind of, and how might that track over time in order to get down to that price?
Brilliant. There are 38 million people in the world today infected with HIV. The incidence of the disease will increase over time because there are more and more people living longer. In Africa, the disease is a disease of men and women. There are more women infected than men. The women are often of childbearing age. They have babies that are infected, and there are more and more babies being born. It's a youth bulge. So I showed a modeling study that I would be happy to talk to you about more. But in the pessimistic scenario that we don't have a vaccine and we don't have better ways of getting out ART in high incidence areas, and transmission continues as it is today, there'll be more people than 38 million infected 10, 20, 30 years from now. So put that into your calculator, right, with respect to the number of doses that might be required for a given vector, and you'll see large volume. This is very different than what is being done by many companies using gene therapy in the United States today, for instance, where the vector is being aimed at an orphan disease, where there are very small numbers of people that have the disease, and therefore the cost has to be higher. We're talking about a high volume disease that requires large quantities of vector. And one of the things I was going to say to the people in the room, I wish I could see you, in addition to reaching out, becoming political, listening to the community, advocating for change, be clever. I mean, I'm an old man, right? I mean, I've seen so much progress in many years of science. Give me 20, 30 more years of being clever. And I loved your statement in the beginning about how you want to help. There's a lot of very clever people in companies that are trying to bring cost of goods down, make things better. And that's, I think something that I'm not being Pollyannish about, but just, I think, being realistic. If there is a need and there is a way, clever people getting together with a mission are more likely to solve the problem than leaving it aside as something that is incomparable.
I would also add, I just want to say, we can't do this without you. Small pharma, big pharma, biotech, we can't. The drugs and treatments don't get to people without that commercialization piece. So you're an integral part of the process. We just have to start thinking together creatively and being clever together to get there.
If we can add, at least in India, volumes is not an issue. I'm sorry.
I take the opportunity to invite that gentleman to join GGTI.
Yes,
so we can have more discussions.
So Sissy and I together would like to say, please reach out. Again, my name is Jenna Dare. You can find me on the app. Anyone is welcome to join the GGTI initiative. and we're happy to have all of you at the table.
So at least in India, volumes is not an issue. I think what is more important is that it's a very price sensitive market. So you have to get the cost down. And when you get there, the bigger challenge is in terms of having adequate capacities to meet that demand.
Thank you for organizing this session around a very essential topic. I'm Nizar Saad from Nationwide Children's Hospital. I have two questions for Mike. So in addition to focusing on HIV and sickle cell disease, is there any plans from the Gates Foundation to bring more gene therapy for LMICs? As we know, like, for example, SMA has been approved and probably other new gene therapies would be approved. So is there any plans to bring those gene therapies to LMICs?
We're focused on HIV, and our ability to work on sickle, which actually was not initially on what they call mission at the foundation, has been facilitated by the fact that the science for sickle cell disease and other hemoglobinopathies is so far advanced relative to that for HIV with respect to curative intervention. And then, you know, of course, the disease burden in sub-Saharan Africa and other parts of the world is so high. It would be unlikely that other indications would be addressed unless they were of high prevalence in low and middle income countries from the foundation, because it has been historically laser focused on high prevalence health issues that have not been tackled by other organizations and that present pathways for solutions. So I wish that we could bring gene therapies for everything, but right now it's the focus is on HIV and sickle.
Thank you. You've answered my second question too.
We have to start somewhere, and HIV and sickle cell are a good start point because of also the very high prevalence. But whatever we develop, I think then creates a platform which we can leverage for other infectious diseases or even non-communicable diseases that are highly prevalent. For example, TB is highly prevalent in our settings, but I think we have to start somewhere and get a solution and then use it to leverage for other diseases.
I should say, if I could add, of the two platforms that I discussed, one relates to therapeutic vaccination, and Sissy's comment about TB reminds me to say that this approach, should it work for HIV, would indeed be applicable to TB, malaria, and other chronic infectious diseases that are prevalent across the world and also in low and middle income countries, and which are on, you know, clearly on the radar screen at organizations like the Gates Foundation. So that's an example where I think it's highly likely that the work that we're doing today to bring what we euphemistically call single shot cures for HIV would be applicable and broadened, not necessarily for hemoglobinopathies though.
Any more questions? Any more questions?
I'll ask one, and this is for Mr. Subramanyam, because manufacturing is challenging, and one, I feel your pain having been involved with vector manufacturing in terms of finding folks who want to be in a clean room all dressed up and can't get to their cell phone every 5 minutes is a challenge. But also, you know, the supply chain issues have really highlighted some of the challenges that we have with obtaining media, our disposables, and the cost of those things really add to the manufacturing costs significantly. And maybe for the whole group, have folks been addressing those kind of issues? I know the gene therapy in a box is one approach, but there's a lot of other pieces that go, and thinking about bringing our costs down, have folks experienced any efforts there to try to attack that part of the supply chain?
Yeah, so I'm glad you talked about some of these points. and I mentioned in my presentation that it's not easy to get good talent into manufacturing. A simple thing like what you mentioned, not being able to use a mobile for a period of time when you're inside the clean room is always a challenge. And we've always debated within the company about allowing mobile phones even in research areas. So we have a very tough policy on mobile use. But more than that, I think there are competing industries as far as talent is concerned. And so the onus is on ourselves to put together good competency development programs. We do offer young development programs for number of areas. So one, we take the responsibility to get good talent, train them. Second, we also get people on a lateral hiring basis. But I think the more important thing is to give people challenging opportunities. You're right in saying that manufacturing is not easy. It's backbreaking to build facilities, to get your all the pieces right. And in the recent past, sourcing of consumables has been a big issue. And then we saw that early on, and we then overstocked as far as inventories are concerned. Even at this time, we are going through challenges as far as consumables are concerned, but I think we've had a reasonable amount of history of engagement with the major suppliers. So we are in that sense in a much better situation. And as far as logistics aspects are concerned in the context of manufacturing, at least in India, we are in a relatively better position because we do have a number of organizations within India. We are able to supply many of the other consumables and chemicals. But if you look at some consumables like single-use bioreactors, we've had a commitment from the vendors, but still you run into problems on and off. And that's a reason that we have taken a combination of both single-use bioreactors and stainless steel bioreactors in our manufacturing. So I think it depends on your strategies as far as procurement are concerned, your strategies as far as configuration of manufacturing is concerned. So all these, and then of course your strategies as far as talent is concerned. So I'm glad you did say that manufacturing is a huge challenge.
Yeah, another question. This is in a really completely different direction. So my company also has a large diagnostics division, and we have a big, well, I don't know about big, but presence in Africa, and we support the H3A initiative. And I've gotten the chance to talk to some of them, and one of the opinions that they've given me was that there's limited resources in these low and middle income countries, and that it's better spent looking at a healthcare strategy more similar to something like South Korea, where you spend a lot of money on diagnostics and preventative medicine, and not so much like the United States and Europe, where you spend a lot of money on pharma, but don't necessarily have as high of a quality of life, which is reflected in a shorter lifespan in the United States relative to like South Korea. So I guess to push back on them, I'm not educated enough to know why their opinion may be missing something, and that there still is room for gene therapies in low and middle income countries. So I'm wondering how you would argue back to somebody who says, no, money's better spent diagnosing because that's been proven to be more effective in different parts of the world.
If it's okay, I can start here. So let me talk about sickle cell disease. Right, identified in 1910 as the first molecular disease. In 122 years, we still haven't brought diagnostics and medicines to Africa. You're right. You know, they don't even know the exact prevalence. Newborn screening's just hitting around 3% penetration right now in the country. They don't have access to hydroxyurea, and a large number of the kids die before the age of 5. We had the profound experience of hearing from Evelyn Musaguera, who is one of our International Community Advisory Board members who has a child with sickle cell disease, talk about her personal story a couple of weeks ago. She is a single mother because her husband left her when their child was diagnosed as having sickle cell because he wanted to go have healthier children with someone else. We have not given them hope. Part of the reason that we don't get a large number of patients advocating, especially in Africa in this capacity, is because there's no hope when they get diagnosed. You bring a curative gene therapy, and we have examples of that in our data here, to the table, and that conversation changes. And by default, you get basic health care that's been missing for 122 years. That is the argument that I would make.
Yeah, in addition to that, I think let us look back at HIV. That was the argument that many people in high-income countries had. You leave low and middle-income country patient with HIV to die, or actually provide prevention and provide counseling, which counseling was counseling people to die positively. And that continued for a long time while we lost so many lives. Now we have seen what antiretroviral drugs have done in terms of mortality, in terms of morbidity, and I think Jane touched on this, even for sickle cell. So we cannot just think, and even people didn't come out to test at the time, because why would you test if there was no treatment? It's because of antiretroviral drugs that people came out to test, because they knew now they had something that they could use to extend their lives. So that argument, I think, was in the past. And even today, if we do not have hydroxyurea for sickle cell disease, but we've got gene therapy to cure those patients, then you don't need hydroxyurea. So diagnostics without treatment may be useful because you could access hydroxyurea if it is available. But still, we are seeing a lot of mortality, both because even when hydroxyurea is there, or we don't have the hydroxyurea. So diagnostics without treatment, I think, doesn't help. So that argument, I don't know whom you're talking to, but I think this has been overtaken by events, seeing what has happened with HIV.
So I do agree that diagnosis is important. But when diagnosed and somebody has a disease, you also need the therapeutics. I remember a conversation with a well-known neurologist who told me that when he tells the parents of a child that the child has DMD, it's like giving a death warrant. So the very little thing that the doctor can do, and that's one of the things that is motivating us to work in these areas. So diagnosis is important, and diagnosis should also be more important in managing a disease risk more than when the disease uh, becomes onset. But at the end of the day, you also need competitively priced and high safety therapeutics or gene or cell therapies to be available, especially for where you have very little options today.
Is there a question?
Yeah, is there
That'll
time?
be our last question. Then.
Okay.
Thank you.
Thank you so much to the organizers, and thank you to all of the speakers. This has been really great. I just wanted to ask about the idea of trust, especially when it comes to clinical trials. We are talking about different diseases and therapies with very storied histories and somewhat problematic timelines. So how do we maintain that trust between these organizations and the patients? The advocacy is great, but even in the broader context, even with COVID happening, there are all of these outside not facilitators, but outside factors that can affect that trust between patients and clinicians and these outside resources. So how can we account for these over time?
So
Conversation,
I'm going to
discussion,
shamelessly plug.
education.
I had to
starting
run
now.
to another session.
It has to happen.
Go ahead, Mike.
I was just going to say what you were probably going to say, Jen. There needs to be communication with the communities that are involved, and we're talking across the spectrum from those doing research to those who are affected by the diseases. And this did not happen, obviously, with the rollout of the COVID-2 vaccines. And look at what happened to that. talking about gene therapy, we're talking about an order of magnitude, if not more, in the way of complexity and probably, chance of fear. So the discussion has begun, even though the interventions are not available, and it has to be made more vigorous over time. Jen, pipe in. I'm sure you have more to say too.
I was just going to do a shameless self-promotion. The session I had to leave for is actually being organized by the Communications Committee, and it was on combating misinformation. I highly recommend you look at the talks, and the talk that I was asked to give in that session was on how to communicate about gene therapy to audiences that are maybe non-specialists. And a couple of high-level points that I just want to make is we all need to learn how to communicate better. And if all of the thousands of people who are members of this society are communicating effectively about what we're doing, not just across our peers, but across every interaction that we have, that information will become a lot plainer to the public sooner. And I want to say, we have to remember, I don't like saying specialist, non-specialist, public, whatever. There is no such thing as the general public. Every human on this planet learns differently and connects with messaging differently. And until we start having those conversations regularly and often, we will not understand what plain language we need to be using. I'll give a great example. Cincinnati Children's Hospital was running a sickle cell gene therapy trial, and they conducted a survey of their patients. And the patients absolutely hated the fact that the word lentiviral vector or even viral vector was used anywhere in the study documents because it made them feel like they were being infected with something. And so the study organizers asked them, what would you like to see? And they said, well, if you explain it to me in a way that I understand it, it sort of sounds like you're not actually using a virus, you're just using the same recipe that viruses use. And it created a plain language exchange where now we say the recipe for is what we borrowed to get the genetic therapy in. And that's plain language that connects with that audience. I highly recommend NIH's plain language resource, which you can connect with online. It teaches you a lot of tricks and tools to communicate more effectively. TED, Technology, Engineering, and Design, has a playlist on how to give a presentation that connects with your audience. And I think you encourage your colleagues to speak more clearly by asking questions. I don't know about you guys, but I've sat in sessions at this meeting as someone who's been in this field for more than a decade, and I did not understand what the speaker was trying to convey. So we don't even talk well to each other all the time. And if we all work on that together and practice that together, I promise you that public trust will come with it.
Thank you.
Yeah, in addition to that, Jane, I think we need to use the people or researchers who are going to work on the patients, for example, in sub-Saharan Africa, to be talking to fellow Africans. So even advocates who are coming from a certain region to learn and be talking to fellow people in the region. And that's why also scientists from developing countries should be part of the development so that when they are talking from an informed position and they can also get trust of the people from their regions.
Thank you. It's been a very good session, and I think in the interest of time, we close the session now. Thank you. Thank you all.